Dear students, welcome to EPG Pathshala. I am Dr. Meena Yadav from Maitri College, University of Delhi. Today I am going to discuss about the nature of hormone receptors. After studying this module, you should be able to identify the major classes of hormone receptors, describe the various intracellular pathways of hormone receptors, discuss the factors that affect the sensitivity of the hormone receptors, describe the mechanism of action of steroid hormones or thyroid hormones, describe the cell signaling through G protein coupled receptors, and differentiate between the intracellular and transmembrane receptors. Hormones are the chemically active substances which, when released in the general circulation, stimulate or inhibit certain functions in the body. Based on the nature of hormones, the receptors vary in structure and the signaling pathways. The hormone receptors are huge proteins, and there are approximately 2000 to 1 lakh copies of these receptors on the target cells. The receptors may be located at different places in a cell, like receptors on the cell surface or inside the cell. Certain ligands like proteins, peptides and catecholamines hormones have their receptors on the cell surface. The receptors can also be present in the cytoplasm. For example, receptors for steroid hormones, and the receptors can also be inside the nucleus, for example, receptors of the thyroid hormones. The number of receptors in a cell varies from time to time. For example, if the secretion of a hormone is downregulated, the number of receptors will consequently decrease, while if the release of hormones is upregulated, the number of receptors will also go up. The properties of hormone receptor interactions. The interaction between hormone and its receptor is well regulated and possesses several characteristics, like the hormone receptor interaction is highly specific. The interaction is carried out in a reversible manner, that is, once the binding of hormone to receptor occurs, it stimulates the intracellular signaling pathways. Once the pathways begin, the hormone dissociates from the complex and is rendered free. The hormone has a very high affinity for its receptor, such that it can bind to its receptor even when the hormone is present in a very low concentration in the plasma. The binding of hormone to its receptors occurs only in the sensitive or the receptive tissue. Talking about the transmembrane receptors, many hormones are soluble in water, like peptide hormones and glycoprotein hormones. These hormones can't diffuse through the plasma membrane. Hence, their receptors are located in the plasma membrane. There are two major types of transmembrane receptors. One is G protein coupled receptor or GPCRs, and second is enzyme linked receptors or kinase receptors. First, we will talk about the G protein coupled receptors. Some hormones like peptide hormones, glycoprotein hormones and amino acid hormones, neurotransmitters and local mediators act by binding to their specific receptors on the target cells, which are coupled to G proteins. These specialized receptors are G protein coupled receptors, which have 7 transmembrane domains, and they are anchored in the plasma membrane. The beauty of activation of these receptors is that the same ligand can activate several different receptor family members. For example, adrenaline can activate at least 9 different G protein linked receptors. Acetylcholine can activate more than 5 different receptors, while serotonin can activate at least 15 different G protein coupled receptors. The G protein receptors are a single chain of polypeptide which crosses the lipid bilayer 7 times and forms 7 transmembrane domains. Hence, G protein coupled receptors are also called as serpentine receptors. How these GPCRs they act? When the hormone binds to the receptor at the extracellular domain, there is conformational change in the receptor, which activates the cytoplasmic trimeric GTP binding protein or the G protein. This results in functional coupling between receptors and the enzymes or the ion channels associated with the plasma membrane. The G proteins in the GPCRs consist of 3 subunits: alpha, beta and gamma. In unstimulated condition of the cell, the alpha subunit remains bound to GDP and G protein remains inactivated. When the G protein is activated due to binding of ligand to the receptor, the alpha subunit releases GDP and replaces it with GTP. Due to this switch, the G protein is then divided into two active components, the alpha subunit and the beta gamma complex. The binding of GTP to alpha subunit causes a conformational change in the alpha subunit such that it releases the beta gamma complex, and the alpha subunit interacts with the target protein. However, the conformation of the beta gamma complex does not change. Actually, in inactive state, the beta gamma complex is masked by the alpha subunit, but in the activated state it is exposed and free to interact with the other set of target proteins. The targets of the dissociated G protein components are either enzymes or the ion channels in the plasma membrane. The alpha subunit of G protein is a GTPase, and when it hydrolyzes the GTP bound to it, it converts GTP into GDP. In this form, that is the alpha GDP, it again binds to the beta gamma complex and forms the inactive G protein. The duration of the switching of GTP to GDP is very short, and it determines the efficiency of the signal transfer. When the isolated alpha subunit GTP complex binds to a target protein or a regulator molecule called as regulator of G protein signaling RGS, its GTPase activity is activated quickly. These RGS proteins act as GTPase activating proteins or GAPs specific to alpha subunit and have important role in inhibiting G protein mediated responses in eukaryotes. In humans, there are approximately 25 RGS proteins. The G proteins are of two types based on their function, stimulatory G proteins and inhibitory G proteins. The stimulatory G protein, the ligands bind to the receptors and the receptors use cyclic AMP as a second messenger, and they are coupled to stimulatory G protein, that is GS. The GS in turn activates adenyl cyclase, which produces more cyclic AMP by acting on ATP. Adenyl cyclase has two catalytic domains towards the cytoplasmic phase of the membrane and two structural domains, each of which contains 6 transmembrane alpha helices. The effect of GS on adenyl cyclase is mainly exerted by alpha subunit, but beta gamma subunit is also involved. Now, inhibitory G protein, also called as GI. This is another G protein which inhibits the adenyl cyclase by regulating the ion channels. CAMP can directly regulate the ion channels, but most of the time it does so by activating CAMP dependent protein kinase A, that is pKa. This enzyme transfers the phosphate groups from ATP to specific serine or threonine residues in the target proteins and thus regulates their activity. pKa consists of 4 subunits, 2 catalytic subunits and 2 regulatory subunits. When cyclic AMP binds to regulatory subunits, the conformation of pKa changes such that the catalytic subunits get released from the complex and get activated to phosphorylate the target proteins, that is transcription factors, followed by the transcription of the target gene. Now, how the somatostatin receptor gets activated? The gene for somatostatin is activated by cyclic AMP. The regulatory region in the somatostatin gene, as in the other genes regulated by cyclic AMP, contains a specific sequence which is called as cyclic AMP response element, CRE. A protein named CRE binding CREB protein recognizes and binds to this regulatory sequence. When pKa phosphorylates CREB on a single serine, a transcriptional coactivator called CREB binding protein CBP binds to it and activates the transcription of the target gene. If the serine has some mutation, none of these proteins can bind and thus it inhibits the transcription even if the CAMP levels are high. The somatostatin receptors are present in the anterior pituitary and brain, and somatostatin can bind to 5 such receptors which differ in their specificities, molecular weight and even the efficiency with which they bind to adenyl cyclase. Once the somatostatin binds to the receptor, it activates the GI protein which inhibits adenyl cyclase. Somatostatin causes an environment of hyperpolarization due to altering the potassium channels, which is cyclic AMP independent. This reduces the intracellular calcium levels as there is decrease in the extracellular calcium influx. Now, mechanism of activation of GPCRs in other hormones. The mode of activation of GPCRs is more or less similar in all the hormones which use G proteins for their signaling. Let us see these receptors one by one. First is LH or FSH receptors. The LH and FSH receptors, they are also G protein coupled receptors, and they act by activating adenyl cyclase and subsequent increase in cyclic AMP levels. The large glycoprotein ligands like TSH or chorionic gonadotropin bind to the extracellular domain of the G protein coupled receptor, while smaller ligands like neuropeptides or neurotransmitters, they bind to the regions within the membrane near the N terminal. POMC, proenkephalins, pro-dynorphin receptors. There are 3 opiate peptide precursors, that is POMC, proenkephalin and pro-dynorphins, and their corresponding receptors, they are named as micro, kappa and delta. Beta endorphin binds to microreceptor, dynorphins bind to kappa receptors, and enkephalins bind to delta receptors with the highest affinity. All of them use adenyl cyclase for the downstream signaling, and all these receptors, they are associated with G proteins. Now, talking about serotonin receptors, there are 3 subfamilies of serotonin receptors, also called as 5-hydroxytryptamine receptors or ST receptors, that is 5-HT1 family, 5-HT2 family, and a family which includes 5-HT4, 5-HT6 and 5-HT7. These are the major receptors of serotonin, and all of them are members of G protein coupled receptors superfamily. The receptors of 5-HT1 family are negatively coupled to adenyl cyclase. The 5-HT2 family works through activation of phosphoinositide specific phospholipase C PLC. The 5-HT3 family of receptors are homomeric and they are members of ligand gated ion channel superfamily, while 5-HT4, 5-HT6 and 5-HT7 are serotonin receptors which are coupled to adenyl cyclase. Next is epinephrine receptors. Epinephrine can bind to 2 types of G protein coupled receptors, that is beta adrenergic receptors, and which are present on the surface of hepatic cells and adipose cells for releasing glucose. In heart muscles where beta 1 receptors are present, they can increase the contraction rate of heart, which increases blood supply to tissues, and they relax the smooth muscle cells in the intestine. Another type of receptors are alpha 2 adrenergic receptors, which are present on the smooth muscle cells of blood vessels in intestine, skin and kidneys. The beta and alpha receptors are associated with different types of G proteins. Beta 1 and beta 2 receptors are associated with GS, which stimulate adenyl cyclase, while alpha 1 receptors are associated with GQ and alpha 2 receptors are associated with GI. The GQ activates phospholipase C and thus acts through inositol triphosphate IP3 and diacylglycerol DAG as a second messengers, while GI inhibits adenyl cyclase. Next receptors are the GnRH receptors. The GnRH neurons express estrogen receptor beta, ER beta, but not other type of sex steroid hormone receptors. The GnRH acts on the gonadotrophs in anterior pituitary via G protein coupled receptors. The activated protein kinase C phosphorylates the serine and threonine residues in the regulatory proteins and initiates the transcription of the target gene. TSH receptors or thyrotropin receptors. TSH receptor is a G protein coupled receptor and it follows 2 pathways of downstream signaling. One is GS adenyl cyclase pathway, another is GQ phospholipase C pathway. TSHR can function either as dimer or as an oligomer, which after ligand binding. The TSHR gene is located on chromosome number 14q and TSHR is the largest receptor in the glycoprotein family of receptors, which includes LH and FSH. The post-translational modifications of TSHR releases two subunits, the alpha and beta, which are joined by a disulfide linkage. Later, the disulfide bonds are cleaved such that the alpha subunit is released, leaving behind the membrane bound receptor. The next category of receptors is the enzyme linked receptors or the kinase receptors. The kinase receptors are single membrane spanning receptors and contain associated kinase enzymes in the cytoplasmic domain. There are further following types, that is tyrosine kinase receptors. For example, they are used by molecules like insulin, epidermal growth factor, platelet derived growth factor. Serine threonine kinase receptors, which are used by Mullerian inhibitory substance and inhibin. Guanylate cyclase receptors used by ANP, cytokinin receptor family, which is used by growth hormone, prolactin, EPO and other growth factors. So, we will start talking about these receptors one by one. First, let us talk about the tyrosine kinase receptors or receptor tyrosine kinases, RTKs. The receptor tyrosine kinases bind to soluble or membrane bound hormones like nerve growth factor, platelet derived growth factor, fibroblast growth factor, epidermal growth factor and insulin. All the RTKs have three domains: a ligand binding site, one hydrophobic transmembrane alpha helix and a cytoplasmic domain carrying the tyrosine kinase. The ligand for EGF receptor is monomeric, while for other hormone receptor the ligands are dimeric. Once the ligand binds to the receptor, it gets dimerized and activates the tyrosine kinases in its cytoplasmic site. The activated tyrosine kinases phosphorylate tyrosines on the other receptor molecule and subsequently all the tyrosines autophosphorylate. The activated autophosphorylated receptor can phosphorylate the other proteins also. After this, the downstream signaling is taken over by the RAS protein, also known as low molecular weight G protein. After this, the downstream signaling is taken over by the RAS protein, which is also known as low molecular weight G protein, which is intracellular GTPase and whose size is 20 to 25 kilodalton, and which switches between active and the inactive forms in the same way as the alpha subunit of G protein. The RAS requires two proteins for its function, that is guanine nucleotide exchange factor GEF and GTPase activating proteins or the GAP. RTKs are not directly linked to RAS, but need the mediator proteins growth factor receptor bound protein 2, that is GRB2 and son of seminless SOS. The SH2 domain of GRB2 binds to phosphotyrosines in RTKs, while its two SH3 domains bind to SOS. This brings the SOS close to membrane bound RAS GTP and facilitates the conversion of RAS GDP to RAS GTP, which is the activated form of RAS. The activated RAS then further carries out the downstream signaling. Now, receptor serine threonine kinases or RSTK family. RSTKs are the transmembrane proteins in the plasma membrane which has three domains: an extracellular ligand binding domain, a transmembrane domain for anchoring in the plasma membrane and a cytoplasmic domain having serine threonine kinase activity. The inhibin receptor is a type of RSTK and inhibin belongs to the transforming growth factor beta family of growth factors. There are two types of receptors for inhibin, type 2 and type 1 serine threonine kinase receptors. Type 1 RSTKs, these are also known as activin receptors or activin receptor like kinases ALKs. Type 2 RSTKs, these kinases phosphorylate type 1 RSTKs. This remains in constitutively active form and is capable of binding directly to the ligand. However, in the absence of type 1 receptor, it can't mediate downstream signaling, characteristic of RSTKs. The ligand binds to type 1 receptor which forms a heterodimer with type 2 receptor. The type 2 receptor phosphorylates type 1 receptor. When the type 1 receptor is phosphorylated, it activates the kinases which are associated with it. The binding of ligand to the receptor causes activation of intracellular SMAD transcription factors which enter the nucleus and regulate the transcription of the target gene. Now, the guanylate cyclase receptors, that is GCs, GC receptors, they are of two major types, the cell surface GC receptor family and the soluble GC receptor family. The guanylate cyclase catalyzes the formation of cGMP from GTP. The ligands for GC receptors are atrial natriuretic peptide ANP and two more molecules isolated with same core structure named as brain natriuretic peptide BNP and C-type natriuretic peptide or CNP. The cell surface GC receptors have three domains, an extracellular domain, a transmembrane domain and a cytoplasmic domain, which contains the catalytic activity at its C-terminal domain. The GC receptors has 7 forms which are named from GCA to GCG. Of these, only 3 receptors, that is GCA to GCC, have ligands that have been identified, while rest 4 are the orphan receptors. The 3 orphan receptors have been found to be expressed in sensory tissues. GCE receptor has been located in eyes and pineal gland, while GCF is expressed only in retina. ANP and BNP both bind to GCA receptor, but BNP binds with less efficiency while CNP binds to GCB receptor. However, all the 3 natriuretic peptides bind to GCC receptor with similar efficiency, as GCC is the most abundant receptor. GCA is a soluble receptor and it has high affinity for nitric oxide. When nitric oxide binds to heme in the cyclase, it stimulates production of more cGMP. The cGMP signaling includes cGMP dependent protein kinase pathway, cGMP gated cation channels and cGMP regulated phosphodiesterase. All these downstream signaling finally leads to the transcription of the target gene within the nucleus. Now, let us talk about the cytokine receptor family. The cytokine receptor superfamily consists of transmembrane proteins which have a common extracellular domain which consists of 2 barrels of 7 beta strands each. The cytokine receptor family includes at least 30 members, and the major ones include growth hormone, prolactin, erythropoietin, leptin, LIF, CTNF, oncostatin M, etc. The mechanism of activation and downstream signaling of cytokine receptors is similar in all the hormones of this family, hence only the GH receptor will be discussed here. The growth hormone receptors. The growth hormone receptor protein is a member of superfamily of transmembrane proteins or cytokine receptor superfamily. There are 2 forms of growth hormone receptor, a full length membrane bound human receptor protein which is 620 amino acids long, having a single transmembrane domain, and it has 3 functional parts: a large extracellular domain, a transmembrane domain, a large cytoplasmic domain, and the growth hormone binding protein GHBP. It is soluble form of protein and is the extracellular domain of the full length receptor. The growth hormone has 2 binding sites for growth hormone receptor. So, when one molecule of growth hormone binds to a receptor, the binding activates the nearby receptor to bind the second domain. Thus, one molecule of GH binds to 2 growth hormone receptors, resulting in a homodimer. This dimerization of receptors activates the intracellular signaling pathway. Growth hormone has multitude of functions in the body and thus uses several signaling pathways to exert its effects. One major pathway through which growth hormone receptor works is JAK2 STAT pathway. JAK2 is a tyrosine kinase and belongs to genus family of cytoplasmic tyrosine kinases. STATs, that is signal transducers and activators of transcription, are cytoplasmic transcription factors. When STATs are phosphorylated by JAK kinases, they move to the nucleus and activate GH regulated genes. A proline rich sequence in the GH called as BOX1 is crucial for the effective gene transcription. Now coming to the steroid hormone receptors. The steroid hormones include glucocorticoids, sex steroids, retinoids, mineralocorticoids, thyroid hormones and vitamin D. All these hormones are small lipid soluble molecules which can easily diffuse across the plasma membranes. The receptors for steroid hormones are transcription factors. Each steroid hormone receptor has 4 important domains, that is a transcriptional activation domain at a minor terminus, a DNA binding domain in the middle. The main function of this domain is to recognize the specific palindromic DNA sequences and response elements for steroid hormones within regulatory regions of the genes. In glucocorticoid receptors, the DNA binding domain contains 2 charged protein sequences which are called as zinc fingers, which have the sequences cysteine X2, cysteine X13, cysteine X2, cysteine. A nuclear localization signal, a hormone binding domain at its carboxy terminus. After the hormone binds to its respective receptor, the receptor is activated and is translocated to the nucleus, and there it binds to cognate response elements on the DNA. This binding regulates the rate of transcription of target genes by either stimulating or inhibiting their transcription. The steroid hormone receptors and the related receptors are soluble in lipids, and hence they can easily cross the plasma membrane. These hormones diffuse into the cell and target the DNA. These receptors include the thyroid hormone receptors, glucocorticoid receptors, estrogen receptors, androgen receptors, calcitriol receptors and retinoid receptors. All these hormones are members of the ligand activated transcriptional factors family. Let us discuss the mechanism of activation and signaling of some of these hormones through their receptors. So, first is the thyroid hormone receptor. Like other transmembrane receptors, the thyroid hormone receptors also have 3 domains. The extracellular domain of thyroid hormone is huge and consists of 300 to 400 amino acids with at least 8 highly conserved cysteine residues. There are 2 major thyroid hormones, T3 and T4. T3 binds to thyroid hormone receptors in target tissues and regulates the release of TRH by hypothalamus and pituitary through negative feedback mechanism. The thyroid hormone receptors are present throughout the brain, suggesting that they play very important roles in the development and functioning of central nervous system. Other than brain, they are also found in peripheral and autonomic nervous systems, bone, muscle, liver, pituitary gland, testis, lung, heart, placenta and some other tissues. Thyroid hormone receptor family. The thyroid hormone receptor is a member of nuclear hormone superfamily. They are just like other transcription factors which bind to a regulatory region in the target gene and regulates its transcription. The gene for human TR is located on chromosome number 14. The receptor is primarily found in thyroid gland, but it has also been located in other cells like lymphocytes, astrocytes, adipocytes, neuronal cells and retroocular fibroblasts. There are several forms of thyroid hormone receptors which have been identified. The 2 major forms of TRs are thyroid hormone receptor alpha, TR alpha on chromosome number 17. The common subtypes of this receptor are alpha 1 and alpha 2, and the thyroid hormone receptor beta, TR beta on chromosome number 3. The common subtypes of this receptor are beta 1 and beta 2. Both of these receptors are derived from different genes and can be alternatively spliced. TR beta 2 is found only in brain, while the other variants are found in several organs. The alpha 1 and beta 2 variants are most abundant in hypothalamus, and more than 80% of neurons in PVN have these variants. T3 binds to TR more efficiently than T4. The hormone ligand complex then binds to DNA via zinc fingers and then regulates the expression of the target gene. Except alpha 2, all other variants of TR bind to T3. The binding of thyroid hormone to its receptor causes heterodimerization of the receptors with nuclear hormone receptors, particularly the retinoid X receptor RXR, which is also a member of nuclear hormone superfamily. And this facilitates the binding of dimerized receptor ligand complex to the regulatory sequence of the target gene to regulate its transcription. The TRRXR heterodimer binds to thyroid hormone response element TRE and regulates the gene transcription. The TR can bind to DNA in three forms: as monomers, as homodimers and as heterodimers with RXR. The normal ligand for RXR is 9-cis-retinoic acid. However, when RXR binds to TR, the complex will not bind to 9-cis-retinoic acid, but instead it will bind to regulatory sequences of DNA more efficiently. Now, talking about the glucocorticoid or mineralocorticoid receptors. There are two highly efficient corticosteroid receptors, type 1 or mineralocorticoid receptor, which binds to corticosterone and aldosterone, and type 2 or glucocorticoid receptor, which bind to corticosterone but with lower efficiency. These receptors are a part of superfamily of ligand activated transcription factors. The DNA binding domains of glucocorticoids, estrogen, androgen and progesterone receptors are similar, consisting of 15 nucleotide sequences, such that all of these hormones can bind to the same region. Talking more specifically about the glucocorticoid hormones, they have receptors which lie in the cytoplasm and they are associated with heat shock proteins HSP70 and HSP90. Once the hormone binds the receptors dissociate from HSP and they get dimerized, which can be either homodimer or heterodimer. After this, ligand bound receptors reach the nucleus, where the zinc fingers of their DNA binding domains fit into the cis acting element called as glucocorticoid response elements or GREs in the DNA. GRE is a palindromic sequence and it has 20 bases. It is also called as enhancer element as it enhances the rate of transcription. They use other transcription factors like NF, kappa B, CJUN and CFOS to initiate the transcription of the target gene. Now, let us talk about the estrogen receptors, progesterone receptors, androgen receptors and vitamin D receptors altogether. All of these hormones have nuclear receptors and they are activated by binding of the ligand. The molecular weight of steroid receptors ranges from 55,000 to 1,20,000 daltons. The receptor for estradiol was the first steroid receptor discovered in the neurons. Estrogen receptors, they have been isolated from adult pituitary, hypothalamus, preoptic area and amygdala. There are two types of estrogen receptors, ER alpha and ER beta. They are expressed differentially and have different signaling pathways. A progesterone receptor from chick has a steroid and a DNA binding subunit and a size of 108 kilodalton. Progesterone receptors, they are found in pituitary, reproductive tract and in most of the regions in brain which contain estrogen receptors. However, they are found in the cerebral cortex of rat which lacks estrogen receptors. Ventromedial nucleus in hypothalamus is the target site for estradiol and progesterone hormones. There are two types of progesterone receptors, PRB and PRA, which are produced due to the alternative splicing of the same gene. Both these hormones share the same inhibitory domain, the DNA binding domain, ligand binding domain and the dimerization domains. Androgen receptors have a size of 120 kilodalton and they are distributed in the brain and pituitary. They are also called as NR3C4. NR stands for the nuclear receptor subfamily, 3 group C member 4. They are nuclear receptors and can be activated by binding to testosterone or dihydrotestosterone, which further activates the downstream transcription factors. Vitamin D is a steroid hormone which is converted to its active form called as vitamin D3, that is 125 dihydroxy vitamin D3 by kidney and liver. Vitamin D3 receptors or VDRs, also called as calcitriol receptors, they are nuclear receptors and contain a hormone binding domain and a DNA binding domain and have a size of 55 kilodalton. These receptors are found in pituitary and brain, especially the forebrain, hindbrain and the spinal cord neurons. The active form of vitamin D after binding to VDRs controls the expression of more than 900 genes which control an array of physiological functions. In summary, the hormones are chemically active substances which can be iodothioronins, amines, small peptides, proteins, glycoproteins or steroids. All the hormones need receptors to show their action. The receptors can be present either on the cell surface, inside the cell, in the cytoplasm or inside the nucleus. The interaction between the hormone and the receptor is highly specific, reversible, has high affinity and it occurs only in the receptive or sensitive tissues. All the hormones can be placed into two major groups, the transmembrane receptors and the steroid receptors. The transmembrane receptors are for those hormones which can't diffuse through the plasma membrane and are further of two subtypes, G protein coupled receptors and enzyme linked receptors or kinase receptors. Some hormones like growth hormone, prolactin, neurotransmitters and local mediators act by binding to their specific receptors on the target cells. These specialized receptors are G protein coupled receptors which have 7 transmembrane domains and hence they are also called as serpentine receptors and which contain a heteromeric protein in its cytoplasmic site called as G protein. The G proteins in GPCRs consist of 3 subunits, alpha, beta and gamma, and due to ligand binding is converted into 2 active components, the alpha subunit and the beta gamma complex. G proteins are of 2 types, stimulatory G proteins which acts by activating adenylyl cyclase and inhibitory G protein which acts by activating cyclic AMP dependent protein kinase A. Somatostatin, LH, FSH, POMC, proenkephalin, prodynorphin, serotonin, epinephrine and GnRH all transmit their signals through GPCRs. The kinase receptors are single membrane spanning receptors and contain associated kinase enzyme in the cytoplasmic domain. There are further following types, that is tyrosine kinase receptors which are used by insulin, epidermal growth factors, platelet derived growth factors, the serine threonine kinase receptors which are used by Mullerian inhibitory substance and inhibin, guanylate cyclase receptors, cytokine receptor family, tyrosine kinase receptors which are used by insulin, epidermal growth factors, platelet derived growth factors, serine threonine kinase receptors, guanylate cyclate receptors, cytokine receptor family. RTKs bind to soluble or membrane bound hormones like nerve growth factor, platelet derived growth factor, FGF, EGF and insulin. All the RTKs have 3 domains, a ligand binding site, one hydrophobic transmembrane alpha helix and a cytosolic domain carrying tyrosine kinase. The second messengers for RTKs are RAS proteins which utilize cGMPs and work in a similar manner as G proteins. RSTKs are the transmembrane proteins in the plasma membrane having 3 domains, an extracellular domain, an extracellular ligand binding domain, transmembrane domain for anchoring in the plasma membrane and a cytoplasmic domain having serine threonine kinase activity. For example, inhibin and Mullerian inhibitory substance use RSTKs for their activity. Guanylate cyclate receptor is a soluble receptor and it has high affinity for nitric oxide. When nitric oxide binds to the heme in the cyclase, it stimulates the production of more cGMP, which acts as a second messenger. ANP uses this receptor for signaling. The cytokine receptor superfamily consists of transmembrane proteins which have a common extracellular domain which consists of 2 barrels of 7 beta strands each. The cytokine receptor family includes at least 30 members and the major ones include growth hormone, prolactin, erythropoietin, leptin, LIF, CTNF, oncostatin M, etc. Growth hormone receptor. There are 2 forms of growth hormone receptor, a full length membrane bound human receptor protein which is 620 amino acids long having a single transmembrane domain and it has 3 functional parts, a large extracellular domain, a transmembrane domain and a large cytoplasmic domain. And the growth hormone binding protein GHBP, it is a soluble form of protein and is the extracellular domain of the full length receptor. One major pathway through which growth hormone receptor works is JAK2 STAT pathway. JAK2 is a tyrosine kinase and belongs to genus family of cytoplasmic tyrosine kinases. STATs are cytoplasmic transcription factors. When STATs are phosphorylated by JAK kinases, they move to the nucleus and activate somatotropin regulated genes. The steroid hormones include glucocorticoids, sex steroids, retinoids, mineralocorticoids, thyroid hormones and vitamin D. The receptors for steroid hormones are transcription factors. Each steroid hormone receptor has 4 important domains: a transcriptional activation domain at amino terminus, a DNA binding domain in the middle, a nuclear localization signal and a hormone binding domain. The steroid receptors are located in the cytoplasm but are attached to heat shock proteins. When the steroid hormones bind to the receptors, the heat shock proteins dissociate from it, causing the receptors to dimerize, which can form either homodimers or heterodimers, which are then translocated to the nucleus. In the nucleus, the dimer binds to hormone response elements or AP1 promoter region at the target site on the DNA and initiate the transcription of the gene. There are 2 major forms of thyroid receptors: thyroid hormone receptor alpha, present on chromosome number 17. The common subtypes of this receptor are alpha 1 and alpha 2, and thyroid hormone receptor beta on chromosome number 3. The common subtypes of this receptor are beta 1 and beta 2. There are 2 highly efficient corticosteroid receptors, type 1 or mineralocorticoid receptor, which binds to corticosterone and aldosterone, and type 2 or glucocorticoid receptor, which binds to corticosterone, but with lower efficiency. Estrogen and progesterone each have 2 types of receptors, that is ER alpha and ER beta, and PRA and PRB. Androgen receptors have a size of 120 kilodalton and they are distributed in brain and pituitary. They are nuclear receptors and can be activated by binding to testosterone or dihydrotestosterone, which further activates the downstream transcription factors. Vitamin D3 receptors, also called as calcitriol receptors, are nuclear receptors and contain a hormone binding domain and a DNA binding domain, and they have a size of 55 kilodaltons. The active form of vitamin D after binding to VDRs controls the expression of more than 900 genes, which control an array of physiological functions. Thank you.